Okay. So yeah, uh, thank you very much for inviting me here, especially Vitor, that was my great mentor in all of the works that we've been doing here. And most of all, we're gonna talk about the protein folding problem that we all discussed during the whole week, mainly going from unfolded structures to native structures using a structure-based model. So this is will be our main tool here. And this is the summary. I'm going to talk about first a story about how quantifying the energy landscape. Uh, we have a great theory behind it with some cartoons that Jose and others showed during this week. And now I'm going to show you a way of quantifying it. The second story, it's going to be about calculating the diffusion coefficient. As Woodside mentioned, it connects kinetics and thermodynamics. And lastly, but we're going to show you some fast contributions to using simplified models. People say, oh, we can apply it, we can apply it, we can apply it. So I did something with some collaborators from my institute in the first couple of years. So what this is the main story that is behind this work. Uh, as I said, many of the energy landscape figures that we show are cartoons that we can describe how that we can discuss how protein folding happens. We can talk about the theory. The theory is all set in this figure. But what we did was to quantify the energy landscape. And for that, what we did was I went to Jin Wang's lab. And at that time, I was Vitor PhD student. And Paul contributes to that. And George Shaini. So these four people together, we work in this team. So as we can see, most people are projecting the multidimensional energy landscape in a two-state free energy profile, where the transition state arises between the two states. But what we can do is also project the multidimensional energy landscape in a one-dimensional representative, where we have all the states discretized, and here is the native states. So as you can see, there is a gap between the states, and we can also project the zero-dimensional energy landscape to the one-dimensional landscape, where we can have the log of the density of the states, which is somehow the configurational entropy. So, well, we have the theory that we can describe. So what we wanted to do is to quantify it. So this is the quantified energy landscape for this protein. And I'm going to show you how we get to this, and how is this related to the other quantities that everybody is interested here. So when I went to that lab, one day I was working in diffusion coefficient, and late in the night, Jin came into my office, and then he started to discuss a lot of things, like how the universe was formed and things like that. And then he asked me, hey, what are the number of the universe? And then he said 42. I had the same discussion with Angel this week. So it seems that people are interested in this number, 42, 42. And then I said, okay, let me think about it. And then I came home, did some homework, and then I said, Jin, I can give you the number of the energy landscape. And he said, whoa, that's a big thing. Can I stop my diffusion coefficient project for a while and do this? He said, well, if you can show me, just prove. So then I went to talk to Vitor, George, Paul Whitford, and Shakun Shum also came in. Now he's in Luis and Vitor Munhoz's lab. He's great on that, in doing simulations in China supercomputers. So yes, what we did was we look at the literature. We found the equations for TF and for TG. Those are related with the, we can find here roughness and the energy gap. And also, it's scaled by entropy. So if you see the literature, people want to try to maximize TF and TG. And that's what we call the principle of minimum frustration. So can we get some quantified information out of that? And the answer with the simplified structure-based model is yes, we can. So in my homework, I came up with this constant, which is in the literature, of course, and that we call lambda. And this lambda is what we're going to do the work for us, which will characterize the energy landscape. So each protein now can be characterized for this by this parameter, lambda. So lambda will be the number that will give you the roughness scaled, the gap scaled by the roughness and by the configurational entropy associated to the landscape. So yeah, the thing is that in the literature, people most of the time they sweep this notation. Sometimes it's big delta E here, sometimes low delta E here. So let's stick with this notation. So I'm going to use this one. So we want to calculate the gap over the roughness scaled by the configurational entropy. So with the theory set, we can calculate TF over TG and see if it has some relationship with these quantities that we have seen so much in the theory back in the 80s, 90s, and 20s, and so on. So we use Paul's code. He contributes a lot during my PhD, during my postdocs, and up to date. He's amazing in sharing everything. And so we calculated the free energy profile for a few proteins. Here you can see a two-state and a one-state protein. And here is one of the plots that we can also make. We can plot the free energy profile as a function of the native reaction coordinate, which is Q, and energy, because the theory relates the density of states with energy. As you can see, is for this type of proteins, they are very symmetric, but there is one application that you will see that's not the case. So let's focus on this. This is where Shakun Shu entered in the project, and then he ran it for a bunch of proteins, including these ones, because we needed lots of samples. We need to set up the density of states calculation in a more rigid way. So as you can see, we have the energy as a function of the density. The density of states as a function of the energy in a 0 dimension and 1 dimensional for proteins of different size and proteins with the same size and different motifs, like the right side of these slides. And you can see that with that in hand, we can calculate the energy landscape. This is a quantified energy landscape, which came out from the theory and from the simulations. So well, after doing this for this group of proteins, we did for a bunch of other dams, a bunch of others. These are proteins with different size. These are proteins with the same size but different topology. So what we asked was, what are the relationship between thermodynamics and kinetics, and can we relate it to the maximization of TF over TG, and of course with the landscape parameter, which is lambda. So we did a bunch of Kinect studies with different proteins in different size, and same proteins, same size. So these are the results that I'm going to show you. There's two classes of proteins. As you can see, there is a relationship between the curvature of the folding kinetics, which are these two behaviors, the first one and the second. And from the theory, the shape of this curve is related with the parameter gap with its small delta E, and the roughness, which is the big delta E. As you can see, this one has a bigger parameter. This one has a lower one, which seems that the gap and the roughness is here, and probably this one folds fast because of this relationship. The gap over roughness is bigger than the other one, which is folds slower and in a more close way. So yeah, here is an approximation, but now we have the exact way of calculating this. These are the thermodynamics for the proteins that I show you. I classified here from two state and one state that came out from the theory, and also I checked with experimental data that we had. And these are the results. As we can see, we can correlate TF over TG as a function of lambda, the parameter that I show you. And as I'm trying to convince you, lambda has this ratio of gap over roughness scaled by entropy. And so we have everything. We have TF over TG, which is a function of lambda. It has a linear correlation, as you can see by the blue line, and you can see the red line is the theory. So both agree very, very well. Of course, that we included non-native interactions. We did a bunch of other experiments, but both correlates very well. And you can see the folding time and lambda also correlates in a linear manner. And we discussed it yesterday about contact order, or absolute contact order. It also has a linear correlation. Okay, so this is a way that we can connect theory by having the topography of the energy landscape, thermodynamics, and kinetics. All of this including in one single parameter, which is the energy landscape, or the lambda descriptor that we've been calling internally. So answering an hell question, the number for your protein is not 42, it's 2.54. So, yeah, that's it. So the second part of my talk is regarding diffusion in protein folding. We talked about connecting thermodynamics and kinetics via the landscape descriptor. Now we're going to connect them by the diffusion coefficient. This is the cartoon of a free energy profile, and we are interested in this quantity, tau f, which is it can be the mean first passage time or the folding time, and it relates the size of the barrier and the diffusion coefficient, which is coordinate dependent. We can integrate in this over Q, and then we have coordinate dependent folding time. We've seen in Michael Wood's side, he put the diffusion coefficient constant, and then we have the Kramer's theory. So it's consistent with Kramer's theory. So as we can, we've seen oh, that was fast. The diffusion coefficient, the diffusion equation, which is a Fokker-Planck equation, can describe the protein folding. Oh, can we? We will try to address this question. And as we can see, the folding time, and I've talked before, it's dependent of the free energy barrier and the diffusion coefficient, and we'll say that the probability of having a configuration, let's say P of Q, is diffusing as Gaussians in this configuration on a space and in time. So this is what the equation is saying. And okay, let's hope it works. So as we are seeing in this plot, the free energy barrier has a component which is dependent of the diffusion coefficient. If we take the log of the diffusion coefficient, we have a kinetic dependence. And for small proteins, this kinetic barrier can arise, but for bigger protein, maybe it's not that much. So we can look at fast folders or barrierless proteins and look for these kinetic barriers, including the diffusion coefficient calculation. So calculate dq is good for looking for this kinetic barriers. So what we did was we implement an umbrella potential in our simulation. So we restrict the configurational space around a Q that we want to calculate the diffusion. And then we can calculate dq in the specific points of the reaction coordinate. So you have here a typical folding trajectory. So we restrict the calculations in this specific Q. And then we have this curve, which is the dq, the diffusion coefficient, coordinate dependent. If we take that in account, you see that there is a small variation of the kinetic dependent thermodynamic barrier and the thermodynamic free energy profile. So we did that for a few cases, and now we are moving on, and we've seen in the literature that there are quite a few of different methods of calculating the diffusion. If you see here, f of q and dq, they have quite similar different shapes, including my own, and mine I say Victor, Paul, and everybody. So what we are trying to do next is see if we can make those methods talk to each other. So what are the parameters that define these? What are the parameters that define this diffusion, and what are the boundaries of this model, and why are they so different among each other? So this is what we're trying to address now. And what we start was with integration of the Langevin equation. So this is a single particle diffusing in one-dimensional coordinate, let's say x. And then we can include in this Langevin dynamics, we can input the diffusion coefficient, let's say dq, and we can input the free energy profile. So we integrate this equation, and we have this sort of trajectory, which is as a function of time. And as you can see here, the input are the black lines. So we input this in the equation, and then we try to see which parameters do we need to use to make the, let's come back again a little bit. What are the parameters for the Bayesian analysis, the stochastic dynamics, or the invariant free energy from these groups? So, and then I'm calling here Hammer is the Bayesian analysis, Sichung-Yang is the stochastic dynamics, and Krivov is the invariant free energy method. I'm not discussing details the method, because what is important here is that we can make them talk to each other. We can extract what are the parameters and the boundaries for these methods to work or not. So for this simple example, it was really hard to make those 4 dq's set in the same equation. So with this, what we did was, what we are doing now is transition path analysis. So what we want to see if the reaction coordinate that we are using is diffusive. The peak of PTPX should be zero 0.5. That is that half percent of the transition that the transition paths are reactive. So it's a diffusive regime, and we are using the Markov theory. So for the single particle that we run the PTP-X analysis, the transition path probability analysis, it seems to be diffusive. It's 0.5. So is it true for proteins and for other systems? So this is what we're trying to answer now. So let's step back a little bit and go to lattice models again, because as we've seen, we can learn physics from this model because they're easy to count states and things like that. So before applying to the real proteins, I'm going to use this model protein. It's a heteropolymer, and it has the properties that proteins has. It folds and folds, and we can have the mechanism, and pretty much we can run in a notebook. So we can test the theory before moving on. So what we are doing is calculating first the thermodynamics for different hydrophobicity cases. Here is low hydrophobicity and high hydrophobicity. The folding shape, the mechanism is different for these two hydrophobicity. I'm going to tell you, okay, this is a lambda parameter that I calculated for low hydrophobicity. Lambda is lower than for higher, which means that we have this different folding funnels for them. And here you can see that for the lower hydrophobicity case, the protein, the heteropolymer, the lattice model, folds in a manner that the correct contacts are forming in the way to the folded state, which is up in the figure. But when we see for the high hydrophobicity case, the heteropolymer collapse. So we have two different regimes that we see in real proteins. So here are two different set of simulations. One is for Robert Best group with all atom simulations from the beta hairpin and the helical protein. And you can see the same trends here for both of them comparing to the hydrophobicity cases. And this is one of our simulations for two-state proteins and downstream proteins that we have this same mechanism. So lattice models were very famous back in the days because at that time they were expensive than the models that you could run in your computers. But now it's good for me because I can test it in my notebook and everything works fine before we move to the real proteins. So the first test, what I'm doing is see if the reaction coordinate is diffusive. As you can see in this plot, this one, it sets in this artifact where we don't have a good probe in the lattice models, but this is where the transition path probabilities is higher. So we are working on that to see if we can find a good reaction coordinate. As you can see, for this special case, the reaction coordinate's not diffusive. I tried to sample and see if I can find for other temperatures, if we can have a peak somewhere close to 0.5, and I couldn't find it. So more work in this direction should be done. And this is the diffusion coefficient as a function of the reaction coordinate using one of the methods that I described before. And as you can see, it has this shape of going up. When we used our model in 2007, we said that the diffusion coefficient goes down. So we are trying to see what are the influences of these two behaviors and if the other methods can tell us what's going on. So this is what we are doing now. This is the results that we had. at that time. And this is the result that we are having now. So if you see the shape of the curvature when you take the diffusion coefficient into account, seems to differ a little bit. The position of the transition state, which here is where it's shown 0.3 kT, shifts a little bit to the right and to the top. And what we are seeing in this previous analysis is that the maximum of the transition path probability is at that point, so we are investigating this now. But what we want to achieve is the folding temperature, or the kinetics connected to the thermodynamics, which is given by this equation. And as you can see, the mean first passage time from lots of trajectories agrees with the folding time when I'm going to call it the analytical folding time, because we calculated with the parameters that we can extract, like the free energy profile, which is pretty much what everybody's have shown in the presentation, but the diffusion coefficient as a function of the reaction coordinate seems to be the key issue to do this calculation. So yeah, this is what we've been doing pretty much in our front, but there are, as I said, people that sometimes in conference and pretty much in my institute when I was hired, they say, oh, you can run simplified models for my system. And then I say, well, let's take a look and let's do some calculations. So I had some contributions in this way. So I'm going to show you just some inputs or some insights that I had with them. So this is the first one. We had some proteins that aggregate, right, set at different conditions. So we have also these steps, maybe it's starting at the intermediate states, and then misfolded proteins aggregate. So the first application was to a protein that does that. And anybody can guess which protein is that? It is the prion protein, yes. So I'm going to talk about a little bit of the prion again. You did the heavy work for me. Now it's just talking about the results. So for the part that doesn't include folding and unfolding, I had to look for another model that I could do this aggregation. And the model is called here normal mode analysis, but with some steps of molecular dynamics. Then we can include not just C alpha beads, but we can do everything that we want pretty much between the boundaries of regular molecular dynamics. So we have molecular dynamics with a rough energy landscape, and normal modes analysis make it smooth, but it's in another set of coordinates. That's why we call normal mode analysis. And this technique does the following. It generates, from an initial structure, we calculate the normal modes, and in the direction of these normal modes, we generate random directions. And these random directions, we explore a little bit the conformational space using molecular dynamics. So this is what we call excited states, and then we let the system relax a little bit. So this is pretty much the results for one protein that we can probe a little bit more NMR structure. So it's a good model for explore a little bit more the free energy landscape around the native structure, comparing to regular molecular dynamics or simple normal mode analysis. So the protein that I was talking to you is the prion, and what we want to see is how the non-infectious structure undergoes to this partially infectious structure. From the literature, we have seen this. It needs to have 40 residues in beta strain, but we achieved only 20, 20 residues in the beta strain condition. So this is what I want to see. We want to see the transition from the non-infection state to the infectious, partially infectious one. So I included the cysteine bound in the structure-based model using the Joanna and Onushik paper. So this is, I was going to show Joanna, but she's not here. Maybe she's listening somewhere. So let's go to the results. This is what we got. We superimpose the normal mode structures on the top of the free energy profile. So with the structure-based model, I can fold and unfold the protein pretty easily. And this would be the most probable path to the unfolded states. But as you can see here, the infectious structures are off path of the unfolding, most probable unfolding trajectories. So if we see at the RMSD deviation, the structure doesn't fluctuate that much. But when we see in the energy, total energy coordinate, you can see that it's sort of a kind of a transition state. So this is what we are investigating now. The infectious structures are not at the most probable unfolding path, but it's off this path. And these structures are sampled at least 1% of the time, so there must be some at least a few KTs to access these structures when we use Boltzmann factor, let's say exponential of the probability of these structures being sampled. So this is pretty much what we're investigating now. And yes, Victor, we can calculate phi values, but computationally, maybe not experimentally at the moment. We've been looking for the literature to see if we can find it, but we couldn't. And what we saw is that these are the amino acids that turn into the infectious state, and you can see that they have lower phi values, which is consistent with this result saying that those amino acids are not native-like amino acids. So yeah, we have this model where we want to see the intermediate states. And quoting Woodside, what we want to do next is to calculate the diffusion coefficient for this reaction, because it's really difficult to calculate the diffusion-dependent coefficient in an experimentally way, and it can give us more information about the transition state of this partially unfolded structure, or at least some insights from the microscopic point of view using theory. So this is what we are doing now. This is what we hope to achieve. We haven't done that yet, but we are. We had a step back, which is when I look for the transition state probability as a function for the reaction coordinate, it never gets 0.5. And now we are working with Paul to find a way of optimizing the reaction coordinate so we can have a diffusive reaction. So far, the Q, it seems not to be, we don't know yet, but it seems not to be as diffusive as we want. If we take this 0.3 seriously, it's going to be fractal diffusion. So we don't know if the regime is subdiffusive. Then we should use other theory. But we are going the direction of using reaction coordinates optimized. Then we can still use the theory that we have, and then we can have diffusive. We can use all the diffusion equation that we set for before. So this is what we want to do. We will include the coordinate-dependent diffusion coefficient and optimize reaction coordinate rates. So this is where we are moving in this in this project. Don't take this DQ seriously now because we have to find an optimized reaction coordinate, but we can see the trends. The trends for these at least is 2 orders of magnitude. So we have to take it into account. I'm trying to convince you that we have to take into account the DQ and not the constant diffusion, at least in a theoretical point of view. So the other example is the Thioredoxin, which is related to cancer and tuberculosis and diabetes and this type of disease. And what we are trying to do is to dock these pharmacophore structures that we determine in our institute at the chemistry department. And what we saw is that it doesn't dock, it doesn't anchor in the PDB structure for this protein. So what we're trying to do is find which structure can we use to dock these proteins. So we run a few structure-based model, and as you can see, there is a region here that's not that sampled that much. So we are looking at this region and try to look at the structures there and propose inhibitors to those structures. So it cannot cause the disease. So this is what we are doing. This is the structures that we select from this region. And then we had a better way of docking these pharmacophores, which has gold on it. So it's thiocarbazone structures, for the ligands. And they have, for that example, they have good affinity and lower, it doesn't kill the mice. So the other fast, not so fast in the point of view of doing that, but a fast application is the human respiratory syncytial virus. We want to focus in the SH protein, where we model that membrane porous, and through the beginning at the structure-based model and finishing at the excited thermodynamics, we modulate the water ion diffusion through the channel, and we found that the histidines, which are pointed to the pores, have a sort of dynamics that select the ion comes in and out. So we can see what are the preferent modes of this selection. It moves like this at the top and like this at the bottom, and then it squeezes the water inside of it, like a pump or an inverse Caesar turning. So this is kind of interesting because we don't have the PDB structure for this system, but we have the position of the histidine using NMR data, so we could correlate the motion with the selection. The other problem which I'm interested in is the Xanthomonas citri, which causes the cancrus citris, which is that yellow thing in oranges and limes. And this is not good for agriculture because when we have it in our fields, in our crops, we have to burn thousands of meters squares, so we lost productivity. So how does that happen? We have this shape for the secretion system of this bacteria, and it's similar to Escherichia coli, E. coli. So we can compare both mechanisms. But as you can see, it's a very complex system. So let's go to a more simplified version of the T4S system, which is big as that. We can see that there is this peptide here around the whole system, and experimental data shows that this peptide has a very strong influence in the way that the pores function. So what we did was to simulate this dimer, and we see that these two amino acids, the tryptophan and phenylalanine, in those positions has the major contribution to the function of the dimer. If we take it off, the T4 system stop to work. And interesting is that E. coli and Xanthonomas citri, they compete with each other, and Shaq is a killing machine. So it kills E. coli. But when we mutate these two residues, it stops killing. So you can see from this gigantic system, we can reduce to the dimer, simulate with structure-based model or regular molecular dynamics, and you can see the influence of these residues of the peptide to the function of the channel. So this is published, but we are moving in the direction of looking at the mechanism of binding and folding that the people in an audience have shown here. But we are interested in the way that B7, which is the peptide, bounds to B9, which is the main protein in the system, and it has relation to the way that the system works. So this is where we are moving on now, and then we want to try to see for the whole system. So this is what we are doing. And in summary, what we talked about was the energy landscape parameter, lambda. I hope I convinced you that lambda can describe the energy landscape for a given protein. And the diffusion coefficient also, it's a parameter that connects thermodynamics and kinetics for the system, and some applications that we can do with these amazing simplified models that the people have been developing and trying to understand quite a while. So this is my group, people who does the heavy work. You can see the guys here. They really do the heavy work. And this is a tree that Vitor gave us. It is Jequitibá rosa, and it can live for 30,000 years. So I planted it. I hope when I'm old, it will be a younger like this, right? So we did this. We did our contribution, and the amazing collaborators that we have. I couldn't put all of them here because selected just the ones that contribute to the work that I showed today. And thank you.